and thank you very much for the invitation to present my work. It's a pleasure to be here. My talk today will be on the evolution of biodiversity across regions, clades, and scales, and perhaps more specifically, on diversity dynamics across scales. One of the most fundamental questions in biology is, how did the diversity of life emerge? When Darwin published this seminal book in 1859, he postulated that all the living things share one evolutionary story. And this story starts with a common ancestor. Now, from this common ancestor, the lineages gradually split over time, generating the diversity of life as we know it today. The whole book contains only one illustration, and it is the illustration of this process. So we have the common ancestor over here, and you see that there is speciation, right? It speciates into 3 species. Over time, some of the species go extinct and others speciate further. Here we see the same process depicted again. So we have a phylogeny, and some lineages over time split, others go extinct. Now, this process of lineage splitting and extinction is called evolutionary diversification. And this evolutionary diversification dictates diversity dynamics. We can describe this process of diversification using the birth-death models. Now, most of us know these models from population ecology, where it is the individuals in a population that either give birth or they die over time. Here, it's the lineages which either speciate or go extinct over time. And we can use the same mathematical framework to describe the diversification of clades on the phylogeny. As an example, we can take the exponential growth equation, which you see over here, and we can isolate the R, right? The R is the diversification rate, which is equivalent to speciation rate minus extinction rate, the birth rate minus the death rate. And by rearranging the equation, you can see that the diversification rate is equivalent to log n divided by t. So this is the log transformed number of species in a clade at present divided by clade age, right? So the size of the clade, log transformed divided by clade age. That's the diversification rate of the clade. So if we have 2 clades of the same age, such as hummingbirds and swifts, which one of them will have faster diversification? What do you think? Which one? Hummingbirds, right? So there's more species, there is faster diversification. You can say that the species accumulate faster within hummingbirds than within swifts. We can take this simple birth-death model and we can extend it to account for time-varying speciation, time-varying extinction. We can also include the logistic growth, so some limitation on the accumulation of species. This is the diversification process, right? So going back to the question, how did the diversity of life emerge? So we agree on the processes of speciation, extinction, and diversification, but what remains controversial is the dynamics of these processes. Right? Like, what are the dynamics of diversification, speciation, extinction, and the dynamics of diversity? While the patterns of diversity in the geographic space across the phylogeny, these patterns of diversity are well resolved, the dynamics that have generated the patterns remain controversial. 2 conflicting views of these dynamics exist, 2 different schools of evolutionary thought, if you will, the equilibrial view and the historical view. The equilibrial view states that the number of species over geological timescales stays more or less stable, right? So the diversity within a clade or within a region is stable over the geological timescales. And it's ecologically deterministic, limited by the environmental conditions. So the environment sets the limit on the number of species within a clade that coexist within a given region. On the other hand, we have the historical view, which emphasizes that the number of species in a clade over time or in a region over time increases. And this accumulation of diversity is highly historically contingent, right? Influenced by sea level fluctuations, plate tectonics, meteorite impacts. All these historical events influence the accumulation of species, which is highly stochastic and dependent on the history of the clade. Now, each of these 2 views has had some very strong advocates, and each of them has been supported by multiple lines of evidence. One line of evidence comes from the biogeography. So the equilibrial view would be supported by the fact that we see universal diversity gradients. We see the same diversity gradients, such as the latitudinal diversity gradient, across many different organismal groups and across different continents, even though these organisms have a very different history. So this would suggest that these diversity patterns eventually converge towards some ecologically or environmentally determined equilibrium. This line of thinking goes all the way back to Darwin. However, it was already Wallace who pointed out the existence of diversity anomalies. Like one of the famous discoveries that Wallace made was the existence of a Wallace's line, which divides very different faunas and floras within a similar environment, within similar environmental conditions, right? So there is an abrupt shift in the composition of the biotas, which cannot be explained environmentally, but it's because of the history of the region, because of the sea level fluctuations. Another line of evidence comes from the fossil record. The famous Sepkoski series, which you probably have seen in the past, shows that the number of families of marine animals has been increasing over the last 600 million years. And you see that this increase in diversity is punctuated by mass extinctions, meteorite impacts, and so forth. So it's a stochastic process, but the number of species keeps expanding. However, recent revisions of this very same data, which you see over here, suggest that after different sampling corrections, the diversity of marine animals might have been remarkably stable over the last 600 million years. And finally, there are some examples from natural history as well. There are famous examples of convergent faunas in different parts of the world, suggesting that the evolution of biodiversity is, to some extent, deterministic, right? Like it's ecologically driven by the environmental factors. But even within these convergent faunas and floras, we often find unique life forms, such as kangaroos in the case of Australian marsupials. And these unique life forms, as the kangaroos, don't have any analog among the placental mammals. So this was the classic evidence: biogeography, fossil record, natural history. But now we have new evidence, which comes from the satellite imagery. We have extensive molecular sequences, which allow us to reconstruct the evolutionary history of a clade with increasing precision. And we can also combine this new data using versatile statistics implemented at supercomputers. So maybe with this new evidence, we can advance a possible resolution of these two views of diversity dynamics. Right? Maybe we can reconcile the two views. But what would such a synthesis of these views, of the equilibrium and the historical view, what would it look like? Today I would like to present an argument that the diversity dynamics might vary systematically across scales. The basic premise is that the dynamics of speciation, extinction, and diversification change as functions of multiple mechanisms involving climate, topography, species traits, and interactions. And all these mechanisms influence diversity dynamics together, but their relative importance varies across scales, temporal scales, geographic scales, phylogenetic scales, producing scale-dependent diversity dynamics. I would like to share with you three studies, three projects which address this issue from different angles. The first one studies the dynamics of diversity, the evolution of biodiversity, across elevations and latitudes. The second one studies the dynamics across the phylogeny, right? So this is in the geographic space, across the phylogeny. And the third one across different geographic settings, islands and continents. Let's start with the first one, which studies diversity dynamics of New World birds across elevations and latitudes. Here we have Humboldt, and when Humboldt was about my age, he was traveling the world, and he noticed that there are many more species in the tropics of South America compared to the temperate regions of Europe. Right? So there are big differences in the diversity at the different latitudes, tropics versus temperate. He also noticed that there are many more species in the lowlands compared to the highlands of the Andes. Now these two patterns of diversity, lowlands, highlands, tropics, and temperate, were confirmed later across different organisms, across different continents, across a variety of montane systems. And they are known today as the latitudinal diversity gradient and the elevational diversity gradient. And these are presumably some of the most common patterns that we see in biology, the latitudinal and the elevational diversity gradient. And because of the ubiquity of these patterns, hundreds of hypotheses have been postulated to explain them. out of these, 3 classes of explanations have become particularly prominent: the energy limits, the diversification rates, and the time for speciation hypothesis. The energy limits postulate that some regions are species-rich, such as the tropics or the lowlands, because they provide a lot of energy for the coexistence of many species, right? So the tropics are productive, there is lots of energy, many species can coexist, compared to the temperate regions or the highlands. The diversification rates hypothesis states that the regions which are species-rich because they accumulate species very fast. The properties of these regions foster speciation, buffer extinction, and as a result, we have more species in the tropics and in the lowlands compared to the temperate regions and the highlands. So it's the rate of species accumulation. And the time for speciation hypothesis finally says that some regions are more species-rich than others because they had more time for the accumulation of species, right? So like the tropics have longer history of colonization and more time for the accumulation of species. Each of these explanations has been supported, but their synthesis remains elusive. In fact, we do not even know what such a synthesis of these 3 explanations might look like. It is possible that all of these explanations apply, but they differ in the magnitude of their effects. It's possible that some of these explanations are more important than others, right? So all of them apply, some are more important than others. It is also possible, however, that the mechanisms invoked by these explanations interact with each other. And the magnitude of effect and the interactions can also change potentially with scale. So we can find differences in magnitude and different types of interactions at the continental scale of latitudes and at the regional scale of elevations. To attempt a synthesis of these 3 explanations, I used the very system that inspired Humboldt in the first place, namely the neotropics. Right? The neotropics are a perfect evolutionary arena within which we see vast areas of the Amazonian rainforest, which is highly productive and provides a lot of energy for species coexistence. In the neotropics also we have a long history of colonization, so there is a lot of time for the accumulation of species. And finally, the adjacent Andes over here might have served as a diversification pump, generating species quickly. So we see a confluence of all 3 explanations: energy, time, and diversification. So within this system, perhaps we can disentangle their interplay. As the model system, I chose 5 groups of birds: the ovenbirds, tanagers, tyrant flycatchers, antbirds, and hummingbirds, which altogether constitute over 1,500 species. And to analyze the data for these organisms, I combined geographic information systems with community phylogenetic structure, which basically studies the relatedness of species in a given region. And under some assumptions, maybe the community phylogenetic structure can tell us something about possible competition between the species in the region. This was combined with diversification analyses, which tell us how fast species accumulate across clades. So these ones accumulate species fast. These ones, the blue ones, accumulate species slowly. The regressions and null models. Let's have a look at the particular predictors, right? So time, in this case, was quantified as the age of the fauna in the region, combined with ancestral reconstructions. Diversification rates were calculated using BAM and DR, and the energy limits were captured in terms of climate and environmental productivity. So now we have the predicted variable, which is the species diversity in a given region, and the predictors. Let's have a look at the results. Here we have the amount of explained variation in species diversity or species richness by time, diversification, and energy. And you see that energy seems to be the most influential factor, followed by diversification and time. And even though time is the least influential one, it still explains about 15% of the variation in regional diversity. So we see that all 3 explanations apply, but they differ in the magnitude of their effect, right? Some are more important than the others. What's also interesting is that when you compare the results for the 5 groups of birds, these relative differences are consistent. So in all 5 groups of birds, it seems that energy is the most important factor, followed by diversification and time. Well, this could be the potential conclusion of the study, right? So we have 3 explanations, and we evaluate them together across multiple groups of birds, And we find that all of them are relevant, but they differ in their relative importance, and this difference in the relative importance is consistent across clades. But perhaps we can go one step further, and we can ask, what are the potential interactions, right? Like, how does diversification change over time as a function of regional energy? Like, what is the interplay of these mechanisms? Let's use hummingbirds as a test case. Here we have the hummingbird diversification shown on the phylogeny, and you see that these two clades stand out in terms of their diversification. That's the bees and the emeralds. Now, the bees and the emeralds accumulate species very fast, as you can see over here, indicated by the red color. And when you look at the map of hummingbird diversity, you see that most of the diversity is concentrated in the Andes. So it might be intuitive to expect that these two groups, the bees and the emeralds, are the Andean groups. But that is actually not the case. So when I project the diversification rates at present onto the map, it will look like this, right? So fast diversification is in North America. These two are North American groups, actually. North America, which is relatively species poor, only recently colonized, but shows fast accumulation of species at present. So it seems that it's the species poor regions which accumulate new species the fastest. Does that make sense? Kind of. Right? We can depict the same results like this, where we have the diversification rate within the region at present and the regional richness within the region at present. And you see that the more species in the region, the slower the accumulation of new species in the region. There is a negative relationship. And interestingly, the diversification also corresponds with community phylogenetic structure. Specifically, in the tropical regions, which show a lot of diversity and very slow accumulation of species, we find phylogenetic overdispersion, which might be indicative of ongoing competition in the region. Right? So there's many species, they potentially compete with each other, which suppresses further diversification in the tropics. In the temperate regions of North America, we see the opposite. There is few species, fast accumulation of species, and phylogenetic clustering suggesting that the species there are closely related to each other as expected under ongoing radiations. And this relationship between diversification and community structure also holds when I account for some null models. So these were the results for hummingbirds. But when we look at the results for all 5 groups of birds, you see that the same results emerge time and again across all 5 families of the birds in the neotropics. So in all of these situations, in all of these groups, you see that diversification declines with regional richness, and this corresponds with community phylogenetic structure. Right? So again, it seems to be consistent across multiple clades. Here we have, as an example, the tyrant flycatchers. Right? So most of the species of tyrant flycatchers are in the neotropics, but the new species accumulate fast. There is fast diversification in the species-poor North American region, where there is very few tyrant flycatchers. So these were the results across different latitudes, right? So at the continental scale, North America, South America. Now let's have a look at the results across different elevations, from lowlands to highlands. Like, what would be the patterns here at the regional scale? Again, we can start with the hummingbirds, and you see that the number of species of hummingbirds declines with elevation. Most of the species are in the lowlands, and as I go high up in the mountains, there is increasingly less species. But interestingly, the diversification, the accumulation of species increases with elevation. So the higher up I am in the mountains, the faster the accumulation of species. And this pattern becomes even more pronounced when I divide the data into North America and South America. Right? So in the species-poor North America, the species-poor mountaintops accumulate new species the fastest. So it's the species-poor regions of the species-poor regions that accumulate new species the fastest. And again, this shows some correspondence with community phylogenetic structure, indicative of possible competition in the lowlands and ongoing radiation in the highlands. These are the results for hummingbirds, but all 5 groups of birds showed the same results time and again. So species richness declines with elevation, diversification increases with elevation, and there is also the correspondence with community structure. So we started with the 3 explanations, and we were thinking about their possible synthesis, right? what would be a possible way to integrate or reconcile these different views of the processes that generate diversity across latitudes and elevations? I would briefly summarize first the patterns. So this is the patterns that we have seen in the data. We saw that each of the three explanations is supported in the neotropics, but their effects vary consistently in magnitude, with energy limits showing the strongest effect. The energy limits are the most important ones. The species richness is high in the long colonized regions, in the tropics and in the lowlands, but contrary to the traditional view, these long colonized species-rich regions show surprisingly slow diversification. And the diversification also seems to correspond with species relatedness or community phylogenetic structure beyond the expectations of the null model. Now that we have summarized the patterns in the data, perhaps we can speculate about a process. Like, what would be a process which would integrate these patterns within a single explanation, within a single mechanism? So the speculation could be that perhaps species accumulate at decreasing rates as regional richness increases, whereby community structure indicates potentially increasing competition. So as the number of species accumulates, this suppresses the accumulation of species, there's increasing competition, and the accumulation of species slows down. Despite these slowdowns, energy does not set a hard ceiling on regional richness. So even the most species-rich regions, such as the Amazonian lowlands, keep accumulating species, right? Diversity is increasing everywhere. But the newly colonized and species-poor regions accumulate new species faster. This interplay of mechanisms that I just described is not captured under any of the three principal explanations, and yet it is remarkably consistent. We saw that it emerges repeatedly across 5 groups of birds, which altogether cover over 1,500 species. What's perhaps even more interesting is that it holds for the latitudinal diversity gradient at the continental scale, right? But also for the elevational diversity gradients at the regional scales. If there were to be maybe one big take-home message from this, is that the tropics do not act as the engine of present-day diversity in birds, but they may have in the past. So in the past, the tropics showed fast diversification, but today it's the temperate regions. So if the tropics act as the engine of diversity, that's a historical function, but today it's the other regions which act as the engine of diversity. This can also be translated into this conclusion that the dynamics of regional diversity were different back then and today, suggesting that the dynamics themselves change with the temporal scale of the analysis. Right? So the dynamics of diversity change over time with the temporal scale. Does that make sense? That was the first study. The second one studies the dynamics across the phylogeny. So we can again start with a question, how species accumulate over time? And we saw that this accumulation of species over time can be captured by studying the phylogenies. This is the very first phylogeny that was drawn, but of course now, decades later, we have very detailed phylogenies, such as this one for mammals, which contain 5,000 species, which evolved over 200 million years. So these are the phylogenies that we now can use as a resource to answer the question, how species accumulate over time. One thing that seems conspicuous when you look at the phylogeny is that the clades differ significantly in their species richness. Some clades, such as rodents, have over 2,000 species, while other clades have only 300 species, or like echidnas, only 4 species. So the dynamics of diversity seem to be different across different segments of the phylogeny. Right? The dynamics of diversity change across the phylogeny. In fact, 4 hypotheses exist about these dynamics on the phylogeny. The first one proposes that the species on the phylogeny accumulate at a constant rate. The second one postulates that the species accumulate at a slowing rate, presumably because of some ecological limitation, or even that the number of species stays equilibrial over time. Some evidence, however, suggests that the species accumulate at an accelerating rate. Right? So we have different hypotheses about how the species accumulate. What we see today is only the number of species in the phylogeny at present, but we would like to maybe better understand their trajectories which led to this point in the present, which will not only help us understand the past, but potentially anticipate the future as well. Right? So if we wait 50 million years, are there going to be 3 times as many species of birds and twice as many species of mammals, as would be the case here? Or is it that there might be different species of birds and mammals, but the number of species will be more or less the same, following the equilibrial dynamics? You can see that this is an important question, not only for the evolutionary theory, so that we can understand the processes that generate diversity, but it seems also key for prediction, management, and conservation, so that we can anticipate how biodiversity responds to the ongoing, for example, climatic changes. To resolve and reconcile the four hypotheses, some of the work, this is the work that I have done, suggests that the dynamics change across the phylogeny, right? The dynamics change across the phylogeny. And to test this hypothesis, I use tetrapod vertebrates as the model system. So that's mammals, birds, reptiles, and amphibians. Altogether, over 30,000 species with very, very live histories. The tetrapod vertebrates also have the benefit that we have very well established and well-resolved phylogenies for each. The previous studies reported different types of dynamics for vertebrates. And finally, the vertebrates span a variety of phylogenetic scales. To study the accumulation of species in the phylogeny, one can use complex methods, such as the diversification analyses, or some of the simpler methods, using tree geometry, for example. The diversification analyses are based on likelihood and Bayesian statistics, which basically fit different types of models into the phylogeny, right? Whereas the tree geometry simply looks at the geometry of the phylogeny, right? So I can use Laplacian spectrum graph theory. I can look at whether the tree is stemmy or tippy. So for example, the stemmy tree has lots of branching events at the beginning, and then the branching kind of diminishes, whereas the tippy tree shows lots of branching toward the present. Maybe I can interpret this as a slowdown in diversification, right? So the branching is fast, but it slows down over time. And this is acceleration. So there's slow diversification, and the diversification, the branching accelerates toward the present. So our two approaches to study the same data. Let's have a look at the results. These are the results for birds, right? And we have the constant rate, slowdown, equilibrium, and acceleration in different colors. And when you look at the colors across the phylogeny, you will notice that the colors are not distributed randomly. So it's not only that the dynamics are different across clades, but they are systematically different. Near the root of the phylogeny, I see mostly red, which is the acceleration, which switches toward equilibrium, slowdown, and constant rate diversification. This is also shown here, right? So this is the time slices through the phylogeny and the representation of different types of diversification. So I can conclude that actually these different types of dynamics operate at different phylogenetic scales. There is a progression. The dynamics change in a systematic manner within birds. When I look at the results for mammals, reptiles, and amphibians, I will find similar results, actually. So for example, in reptiles, we also see that there is acceleration, equilibrium, slowdown, and constant rate diversification. So it seems that there is a change in the dynamics with the phylogenetic scale of the analysis. You see the same results depicted here in a different way. So that's the size of the clade, acceleration, equilibrium, slowdown, constant rate diversification. So small clades show constant rate diversification, large clades, acceleration, and so forth. Tree geometry, which was the other type of analysis, the tree geometry supports the same trend. These are the results for birds, right? So we see that the geometry of the phylogeny, the topology, the branching times change with increasing clade age and clade richness. And again, I find the same change, the same trend for mammals, reptiles, and amphibians. So it seems that the trend itself is very well established. Maybe we can speculate about how to potentially interpret this trend biologically. So what do these results mean? It might look potentially something like this. So this is the number of species which accumulate over time. And I see that within the shrews, bats, and rodents, the species accumulate at a constant rate, at a slowing rate, and eventually there is an equilibrium, right? So the rodent niche is eventually filled with rodents. The bat niche is filled with bats. However, the mammal diversity keeps increasing, presumably because there are some key innovations which unlock new niches, new ecospace, new opportunities for diversification. So even though I see these different types of dynamics, they all actually fit together. there is a way to propose a synthesis of these different types of dynamics. You see that even though this makes potentially sense biologically, it's not the kind of synthesis that would occur to us probably from the outset. This could again be the conclusion of the study. So we investigated the 4 hypotheses. It seems that all of them apply. They're not contradictory. They actually complement each other. And the key ingredient to their synthesis is considering the scale of the analysis. But maybe we can take this again one step further and look at the potential causes of these patterns. So why do we see these patterns in the data? We were able to come up with 4 potential causes. And typically, we use the biological explanation, right? But today, I would like to focus on this one, the survival bias. The survival bias refers to the fact that only some deviations from the random branching process generate a given observation, right? So let's imagine that I simulate a phylogeny. Let's imagine that I simulate 1,000 phylogenies using the same speciation rate and the same extinction rates, right? So like I simulate 1,000 phylogenies under the same speciation and extinction rates. Now, these phylogenies will be different from each other because it's a probabilistic process. Most of these simulated phylogenies will look something like this, right? There's the random branching, the lineage is split and go extinct over time. I get a phylogeny like that. But because I generated 1,000 of them, a minority of the phylogenies would show the late branching or the early branching, because simply by chance, there was a lot of branching early on or a lot of branching later on during the simulation process. Now, the interesting thing emerges when I try to simulate very large phylogenies with thousands of species, right? So if I simulate phylogenies which contain 10,000 species, 5,000 species, I will find that most of the phylogenies will look like this, showing the early branching. Now, the early branching is not really representative of the simulation process. It's a biased sample of all the phylogenies that were produced. But because there was lots of branching events early on in the simulation, the phylogenies became big very quickly and had a higher chance of survival all the way to the present, right? So the early branching increases the chance of survival over the long term all the way to the present. And I get phylogenies which show the early branching. Does it make sense? Right? It's not a biological effect. It's simply a systemic selection, right? Like only certain trajectories survive all the way to the present, especially when simulating large phylogenies. So what we can do is that we can actually investigate the survival bias by running all sorts of simulations, and we can go back to this in case you would be interested. But basically, here we have the empirical results that you have already seen, acceleration, slowdowns, equilibrium, and so forth, and the geometry of the phylogeny. These are the empirical results for vertebrates, and these are the results of the simulations with the survival bias. And you see that the survival bias itself remarkably reproduces what we see in the empirical data, right? So even without considering any biology, using just the survival biases themselves, I can actually replicate closely to what I see in the data for mammals, amphibians, reptiles, and birds. I would conclude that the trends in the diversity dynamics do not necessarily require a biological explanation, right? They might result for non-biological reasons. Just to briefly summarize, we saw that phylogenies show previously unknown trends from constant rates to slowing, equilibrial, and accelerating diversification. These trends can reconcile the previous results and the conflicting dynamics. And the trends seem to be largely non-biological, consistent with survival biases. The biology can still contribute to the observed trends to some extent, but it seems that we do not necessarily need it to reproduce the trends. Perhaps the most fundamental conclusion here is that the large phylogenies principally differ from the small ones, right? So the large phylogenies have different geometry, different topology, different branching time distributions than the small ones. And this has potentially nothing to do with the biology itself. It's simply built in the process of how the phylogenies survive over time. So if I basically analyze a large phylogeny and a small phylogeny, I expect to get different results. This is potentially relevant for multiple fields which use phylogenies in their research, such as the phylogenetic comparative methods, conservation phylometrics, community phylogenetics, where different phylogenetic scales can support different conclusions. So if I have my question, my hypothesis, and I choose to work with hummingbirds, birds, and vertebrates, I might expect to get different results simply because the phylogenies for these clades have different shapes and sizes. In ecology and evolution, we widely consider the temporal and spatial scale. Those are very broadly used. But the results that we have just seen also underscore that maybe the phylogenetic scale might be relevant to consider. This is a concept which we have conceptualized and developed in a paper that followed from my dissertation, and that's a conceptual study. It's conceptual arguments. The results which I just showed you might provide some empirical arguments, some empirical evidence that scale might need to be more considered so that we can make sense out of these seemingly conflicting patterns. So how do species accumulate over time? Well, we saw that the diversity dynamics might change across phylogenetic scales. And this brings me to the last study, which focuses on diversification across islands and continents. Sorry. For this study, we used corvid passerines as the study system. The corvid passerines are these charismatic birds which you see over here. It's about 800 species from 30 different families. And we know some corvid passerines also from the Czech Republic, such as the crows, the ravens, the magpies, from the family Corvidae. But the corvid passerines also include, for example, Madagascan vangas, which are known for the spectacular radiations which unfolded in Madagascar. There is also the monarch flycatchers, or the birds of paradise, right, with the sexual selection, conspicuous feathers. So all of these are the corvid passerines. The corvid passerines are distributed around the world, as you can see over here, but the biodiversity hotspot for these birds is in the Indo-Pacific. And we have very detailed information about the distribution of these birds across more than 1,000 islands of the Indo-Pacific. So this is a very detailed data set. We have information for both the island systems and the continental system. Now that we have the same organisms distributed across islands and continents, we can ask, what are the dynamics of diversity across islands and continents? Are these dynamics similar across these different geographic settings, or are they principally different? And if they are different, in what specific way? Let's start with the islands. We will have two predicted variables, which is island diversity and diversification. Island diversity is simply the number of species on an island. Island diversification is the average diversification rate of the species on the island, right? So this measures how fast the species accumulate at the island and how many species are on the island. We can go back again to some classic theory for guidance and potential predictions. MacArthur and Wilson introduced their theory of island biogeography, where they argue that the number of species on an island, the island diversity, results as an equilibrium of island colonization and extinction. So these two processes eventually equilibrate, which will give me the number of species on the island. But even early on, this theory of island biogeography was criticized by David Leck, Daniel Simberloff, and others, who argued that this equilibrium model should be seen as a useful null model, but not really as an empirical expectation. In fact, because of perturbation, dispersal limitation, plate tectonics, or sea level fluctuations, most islands are supposed to be outside of their presumed equilibrium, right? So this is just a null model. The empirical reality is more complex. And of course, there is other processes beyond colonization and extinction involving geographic, climate, and so forth. And we can use these mechanisms to generate specific predictions. For example, we can predict that the geographic configuration of the islands might facilitate speciation and suppress extinction. The mountains on the island can generate diversity and serve as refuge from extinction. We can also predict that climate, especially temperature, sets the rates of population divergence and speciation and determines the ecological limits on island diversity. Niche position, partitioning, and pecking can further influence the diversification process. And finally, competition has been hypothesized to suppress diversification, right? So the more species on the island, the more they compete for resources, and this suppresses the accumulation of species. To capture each of these predictions, each of these mechanisms, we can use specific variables. So I captured geography using island area and isolation, island topography. Climate was captured using temperature, precipitation, environmental productivity. Traits using functional volume, evenness, and divergence. And finally, I also used community phylometrics. We saw this before. Of course, competition is impossible to directly study based on correlative data. But maybe we can make some assumptions and see if our predictions are fulfilled, right? So like if I predict that the community phylometrics indicate competition, is that connected with suppressed diversification? Let's have a look at the results. You have island diversity, island diversification, and the amount of explained variation by each of the 4 classes of predictors. The first thing which stands out is that these two, island diversity and diversification, are not correlated. They're decoupled from each other. And each of them seems to be governed by different factors. So island diversity is determined primarily by island geography. The effects of climate, traits, and community structure are limited. Island diversification, on the other hand, is largely independent of geography, climate, and traits, but there is some correspondence with community structure. Let's have a look at the results in more detail, and we will see that the number of species on an island increases with island size, with island area. And the island area alone explains 55% of the variation in island richness, right? So the area alone seems to be a key predictor. What's perhaps even more interesting is that when we look at the points below or above the line, right? So these are islands that have more or less species than expected for their size. The islands that have more species than expected for their size, right, these ones, show significantly suppressed diversification, right? So there's many species, more species than expected for an island of given size, and there is suppressed diversification on the island. We also see the opposite. Islands that have less species than expected for their size show significantly increased diversification. So they accumulate species fast. Those would be the islands below the line. These islands are typically the old continental islands, the large ones, whereas these islands with diversity deficit, increased diversification, are small and isolated islands which are young, such as Fiji and Vanuatu. This would be like Borneo and Sulawesi. There is also an interesting correspondence, again, between diversification and community phylogenetic structure, where we see overdispersion being coupled with suppressed diversification, clustering with accelerated diversification, and these seem to constitute multiple lines of evidence for the equilibrium dynamics of island diversity. So we saw that island diversity and diversification are decoupled, governed by different factors. We saw that island diversity is deterministic, governed by island area, and island diversification correlates with proxies for possible competition and with proxies for niche filling following from the species-area relationship. So each of these lines of evidence would be relatively weak on its own, but when you look at them together, there seems to be some indication that the dynamics of island diversity might be equilibrial. Now let's move to the continental system, right? It's the same organismal group, but this time we're on the continents. And you see that most of the species of corvoid passerines are distributed in the tropics. However, there are significant differences between Southeast Asia, Africa, and the neotropics of South America, right? So the diversity of corvoid passerines does not really correspond with environmental conditions. How do we explain this? Like, what could be the other factor besides the environmental conditions? We got an answer when we looked at the phylogeny and reconstructed the historical dispersal of corvoid passerines around the world. By looking at the dispersal reconstructions, we will see that the corvoid passerines originated in Southeast Asia, and then they spread into Africa and through Asia and North America into South America, right? So the Southeast Asia has been colonized for the longest time. There is most of the species. Lower diversity is in the later colonized Africa, and the lowest diversity in the most recently colonized South America. I can also plot the results like this, where I look at the number of species in a given bioregion or biome and the time since the biome or the bioregion has been colonized. And you see that this time of colonization in itself explains 58% of the variation in biome richness. Right? So it's not the climate of the biome or the size of the biome, it's really the history, it's the timing of the colonization, it's the history itself that explains a lot. We can also compare the effects of time to climate, geography, topography, functional composition, phylogenetic composition, and again, time seems to be the most important factor. We can also investigate the functional diversity, right? So this seems to suggest that the corvoid passerines accumulate at a constant rate around the world, and as they colonize new region, they slowly accumulate species in a clock-like fashion. So there's continued diversification. And maybe there is also continued divergence in terms of functional diversity, which seems to be supported by this scatter plot, right? So functional diversity increases linearly with regional species richness or regional diversity. So even the most species-rich regions, such as New Guinea, don't show any sign of saturation, right? The niches being filled with species. All of this supports the expansionary view. Diversity is expanding. It depends on the history of the region rather than its environment. I would therefore summarize that the island systems seem to be equilibrial. The size of the island determines island richness. Potentially, competition suppresses diversification, and diversity converges on an equilibrium. The continental systems follow very different dynamics. So here it's really the environmental factors, the size of the island, that determine diversity, deterministically. Here on the continents, it's not the environmental factors, but the historical factors, which determine biome richness. Specifically, biome richness is determined by the timing of dispersals. There are no signs of functional saturation or ecological limitation, and diversity seems to be expanding everywhere. The species accumulate in a clock-like fashion all over the world. This suggests that the diversity dynamics vary across different geographic contexts, islands and continents, and potentially across geographic scales. We saw 3 studies today. The first one looked at diversity dynamics across elevations and latitudes, across the phylogeny, and across islands and continents. Here we saw that the diversity dynamics in the past and in the present are different from each other, right? The diversity dynamics change with the temporal scale. Here we look at the diversity dynamics across phylogenies and clades of different ages and sizes. And we saw that the small phylogenies are principally different from the large phylogenies. They have different geometry, different branching time distributions, different topology, which is consistent with different types of diversity dynamics. So the dynamics seem to change with the phylogenetic scale. And finally, we saw the comparison of islands and continents. Whereas on islands is the regional environment, the island size, which determines diversity. On continents is the history of the region, the time for species accumulation that determines diversity. So it seems that these dynamics of diversity might change between the small scale of the islands and the large scale of the continents, varying with the geographic scale. So how do you reconcile these two views, the historical view and the equilibrial view? The historical view stated that species accumulate over geological time, and this process of accumulation is dependent on the history of the region, evolutionary and historical contingencies. The equilibrial view states that the number of species is very stable over time, determined by environmental conditions. And it seems that based on the results that we have seen here, that both of these views are valid. Both of them apply. But they apply at the different scales, geographic, temporal, and phylogenetic. So scale maybe is one key ingredient which can reconcile these two views. And when we consider the scale, these two views appear not to be conflicting, but they actually complement each other. So the different views are valid, but at different scales. And maybe when we consider that the diversity dynamics might vary systematically across scales, we will be in a better position to uncover the interplay of mechanisms which together generate diversity in the geographic space and across the phylogeny, so that we can advance the resolution of some of these classic questions that have fascinated biologists ever since Darwin, Humboldt, and Wallace. Thank you.
Yeah, thank you for this great presentation. It was dense, and I have to go over it several times to digest all the details which were there. But still, I am convinced that there will be a couple of questions. I see his hand is raised. Then Peter.
I have many questions. I have many questions, but I will start with a very simple one. These two guys, which you showed us all over again, you withheld their identity from us. I'm sure I've seen the faces before, but could you finally disclose their identity?
Does anybody know?
Many surely don't.
This is George Simpson, which was one of the architects of the evolutionary synthesis in the 20s, 30s, and this is Ledyard Stebbins, who was the botanist, I think in Stanford or in Austin. One of those. So that's Stebbins and Simpson.
Okay, so Peter.
Okay, we are on a botanical seminar, so I would like to ask.
No, it's a joint seminar.
Joint seminar, okay, but okay, it's the time and place, so.
No, it's a joint seminar.
Okay, so how will be this with the plants? Do you expect that the patterns will be absolutely the same or different? And why this is not studied primarily in plants but in animals? So what are the advantages of animals, and what do you expect will be the advantages of plants for studying the same?
I feel like one of the limitations with the plants is that, and maybe you can correct me, right? Like, we don't have that accurate information about the phylogenies. That seems to me like one of the limiting factors, and the geographic distributions. So in that case, I feel like this would be a perfect opportunity to test some of these ideas. And like some of these ideas go back decades or even hundreds of years, to test them using plants. Because in principle, there is nothing specific to these hypotheses, to the organismal system. Right? So for example, the diversification rates in the tropics that might apply as well as in amphibians, also in plants. So it would be an interesting comparison. And like as we get more and better data, that would be great to do.
Thanks. Maybe I will actually continue this question and further develop, because it's interesting that in plants, if you look at this relationship between energy, or let's say resources, and species richness, we mostly see a humpback relationship, because of competition, which is probably a stronger force in plants than in animals because of their sedentary life form. Yeah, so that may be one of the big differences you may expect.
So I guess the question is going toward like the patterns of diversity that we see are different, right? It's hump-shaped versus linear. Yeah. My intuition, and again, like you know more than I do, I feel like it also depends on the scale of the analysis, right? So like at the local scale, the diversity-productivity relationship is hump-shaped, but when you look at the global scale, most of the plants would be in the tropics, which are the most productive region, right? So as long as the patterns are the same, we can use the same hypotheses, and maybe even if the patterns were different, it could still apply that there is more diversity in a given region because there is maybe not more energy, but there is faster diversification or more time for the accumulation of species.
Yeah, I would agree, yeah. My question is about extinctions, because you used the first study, you spoke about the diversification rate. It is, I think, about the recent species pool, but there should be a lot of extinctions which should affect the results. So how it is included?
That's one of the big challenges, right? So typically, we can circumvent this by not studying speciation and extinction separately, but we can study them together. That's what we call evolutionary diversification. So we don't know how much of the diversification can be attributed to speciation or extinction. And another way to make sure that the results are less impacted by this issue is to focus on present-day diversification. So we can estimate with high confidence what were the diversification rates near the tips of the phylogeny, like the last 2 million years, basically.
Sorry, and then it will be your turn. But as you showed that the tree, the phylogenetic tree has some balance, or the structure of the tree could be expected, that could you then somehow evaluate the number of extinctions, because you can see the difference of your tree to the random, which you can produce by the system?
Definitely, and this has been studied using mathematics, right? So on one hand, we have the equations which tell us the birth test process, like what would be the distribution of the branching times under the assumption of constant rate speciation and extinction, and we can show that these mathematical equations, they work. They work well when applied to the simulated data, where the assumptions hold. The issue is that, in real biology, the assumptions rarely hold, and that's why it's problematic. So we can infer the extinction in principle, but with the real data, it gets more difficult because the assumptions don't hold.
Thank you. I have more, but it's your turn.
I also put forward just one in the beginning, so now another one. You shortly mentioned the theory of island biogeography, and I'm sure that when Wilson and MacArthur wrote about that, they were well aware that it was very simplistic. I think they even wrote that. Still, you've shown us later that the size of the island is indeed very determining when it comes to species richness of an island. And just my question is, as you've shown that, it would be natural to test also for this distance from the big species pool, from a mainland or a big other island. So did you do that, and what was the result of that?
Definitely, and I didn't mention this, but basically I tested both, right? The distance and the size of the island. And when I was talking to people who work in the museum and they know these birds really well, they would say that these birds are dispersal limited. They cannot get far away. They cannot fly across the Indo-Pacific. Like, obviously, the island isolation will be important. But when we looked at the results, and that's where they were surprised, it was really the size of the island, not the distance of the island from the other islands or from the mainland. So in this sense, on one hand, this equilibrial theory of island biogeography was supported. Area is important, but for these birds, actually the distance doesn't seem to play a key role.
And students, if you want to ask, you have to be fast, because we have a lot of
Okay, I would have a question to calculation of diversification rate. Could it be somehow biased by some taxonomical bias? Okay, okay, if a group has so many species, people try to be lazy to describe new ones, or it's already enough, and so on. Because if I imagine that this will be the case, this could easily explain your first paper. Yeah, that okay, in a region when you have only 2 species, people are very careful in describing these 2 species and distinguishing. But if you have 100 of birds somewhere, it's perhaps too enough people to learn and even to describe more. So, do you expect that there will be such a kind of bias? And have you some evidence that this is not the case?
I think that the bias will be there. Like, when we think about it, so there's more species in the tropics, but maybe there is actually even more. We just do not differentiate between them because the tropics are not as well explored. And you're asking, like, maybe if we actually distinguish the species in the tropics, the diversification would be faster in the tropics than it seemed from these results. So that's a possibility. Like, my intuition is that we know enough for the clades in like the last 2 million years, like to do the distinctions so that we cannot overturn these results that we saw here. Like, the differences are too big to be explained by these bias, but it's difficult to quantify the bias itself rigorously. So that's a great point, and it should be discussed as a caveat because it's problematic.
Could it be the way that somebody, let's say, sample 1,000 of birds, sequence them in a region randomly, and will divide not by the taxonomical ranks, but let's say by the diversity of sequences, whether the diversification is faster here or not? Not to introduce some personal bias. This could perhaps function in this way.
It would work, and maybe it would be an interesting additional test, right? Because we cannot do this for all the species of birds, but maybe selecting a clade where we have the sequences for enough species to test for the genetic diversity and the genetic divergence rather than the number of species. That's a great idea, absolutely.
Yeah. About your last talk, you mentioned that even before, I mean, of course, I think it came to my mind and probably to the mind of many others that there should be this niche saturation, niche filling. But you talked about corvoid passerines, right? And you made some conclusions based on them. Of course, it's not a small group of birds, but isn't it so that by expanding, for example, their range, colonizing new regions, then speciation would occur, that they would expand, also in terms of species and filling niches, on the expense of other birds, and maybe not even just birds, but bats, you know, when it comes to nectar on flowers and so on. And would that not lead maybe to false conclusions because you don't look at the whole picture?
Definitely. You know, like, I feel that the question is like, what is the natural unit of analysis? Is it corvoid passerines? Is it birds? Is it vertebrates? And that's a difficult question, right? Because we never really know. One argument which I would put forth for the fact that these results are meaningful is that I can actually predict the number of species of corvoid passerines on an island if I know the island size. So if the corvoid passerines were not a meaningful natural unit, and in fact they were competing here with bats, here with other birds, I would not be able to predict the number of species because it would not really be the number of species of corvoid passerines, but the combination with the bats and the others. And we cannot explain 100%, but 55% of the variation is a lot, and it seems like they're actually meaningful unit.
Okay, yeah, Gonzalo.
Thanks for the talk. I truly loved it. I have several questions, but I will try if I can make two. The first one would be if, you know, in the first study of the passerine birds, have you considered the distance to the origin of a speciation, if this might be some of the, the farther away, the fastest is the increasing the diversification rate, as it is in a, we suppose, more distant environment, so more opportunities to explore new niches. And yeah, so an invasive species is not the same if it crosses the Strait of Gibraltar than if it crosses the Atlantic Ocean. Well, perhaps it's actually the opposite, but. Okay. And the second question is, I've just read somewhere about analogies between the R and K life strategies and the diversification rates that clades which tend to diversify very fast also tend to have much stronger extinction episodes, while there are clades which diversify slowly but are more resistant to sudden decreases of diversity. So have you ever crossed about this, or do you have some opinion on that?
There's this life and slow life histories as well, which is kind of R and K, and the idea would be that the clades with the fast life histories, like rodents, would show fast speciation, their speed of life is faster, they have more generations, so in the actual physical time, they have more generations, they have more speciation, and that's why there are more species of rodents compared to Afrotheria or the primates. So it's possible. I don't think that I have seen any study that would actually confirm this rigorously, but it's a... Ah. There are implants? Interesting. So you're saying that fast speciation is also coupled with fast extinction. That's interesting. Like one comment which comes to my mind is, for example, when you have clades with small ranges, right? Those small ranges are prone to speciation, but also prone to extinction. So there are mechanisms which increase both the rates of speciation and extinction at the same time. So definitely it can happen, and it seems that in plants it's the case.
Thank you for these questions. So are there another? So, yeah.
I would like to ask, you've talked there about the elevation diversity gradient, and I would like to ask if it was somehow corrected to the area or not, because I think that we have quite a lot of lowland, but only somewhere are some mountains or hills, and if it will somehow change, if we do some correction like that for like same plot or not. Yeah, thank you.
Definitely. In this case, the units of analysis were always grid cells, which is 100 times 100 square kilometers, so they were of the same area. But I feel like you're saying basically that the energy hypothesis and the area hypothesis are somehow connected because there's more energy per area in the lowlands, which could also explain why there is more species generally. And the area itself can also facilitate speciation and protect the clade from extinction, right? The bigger areas, allopatric speciation and so forth. So it would be interesting, to investigate it and to add area into the models, because now we have just energy, time, and diversification, and I feel like the area would fit within the energy hypothesis and could be added. Good idea.
I have another one. No, two. Could higher radiation in high elevations, well, it should increase mutation levels. Could that kind of have an effect on speciation, which would be measurable? One thing, and the second one is a stupid one. But I wondered all the time, why did on most of your graphs the southern tip of South America disappear when zero should have been violet?
So that's like a plotting question, but it's kind of like about the radiation. Yeah, you mean the UV radiation increases the mutation rates and this facilitates diversification. That would be interesting to look into, right? I have no intuition for how much of the radiation there is and if it could account for the differences. But it's an interesting idea. That's for sure.
You will not explain South America.
It was zero.
Okay. So, yeah, Michal and then Kuba.
Hello. Thanks for a nice talk. Yeah, it was mind-blown. I mean, in a couple of times my wheels were stuck, and it's weird to talk to the microphone. And if I get it right, there was a negative relationship between diversity and diversification, and you also presented that larger phylogeny tend to look like with long branches. Was it right? If you simulated them, but also it was something that you can see in nature. So I was thinking how much this is kind of a mathematical or ultimative artifact that it can happen other way around. And so it means if you have all the diversification, there is always a smaller diversification rate, so more species, and we always see lower diversification because there is no other way around. Can you just produce the opposite? Or, I mean, I have a feeling that basically it's impossible, so it has to ended up that way. So what do you think?
I think you're right. That's a great insight, right? Because you're saying it's just the structure of the phylogeny itself dictates that the small phylogeny, such as the temperate ones, will have faster diversification.
Mass extinction. Bigger
yes.
phylogeny holder has much probability to extinct. So I mean, if you cut the phylogeny, it's much better to speak without the microphone. So if you take the bigger phylogeny
I can see the connection, and it's a good insight. I feel like maybe one way to implement this would be to actually study diversification for the simulated phylogenies, which I haven't done, but it could be interesting to have a look at it, like to see, like
You can see
this could
something
be
else, or this is just the automatic output
absolutely.
to how phylogen.
Definitely, I agree.
Actually, I would like to return back to this energy hypothesis and plants, because actually what we can see in plant diversity patterns is not the highest diversity in tropical lowlands. Yeah? Instead, actually, there is more diversity in tropical mountains, and we also have like remarkable biodiversity hotspots in mountains areas like worldwide. Yeah, so basically, these patterns which you actually showed for birds and so on doesn't really hold for plants. Yeah.
I guess, when you look at the research for the type of bird, you see that it's in the tropics. The most of the diversity is also mountain. The mountain areas of the end. So there's definitely differences, but there are also some surprising similarities, given that the plants and the vertebrates, they respond to the environment surprisingly similar.
Yeah, on the other hand, that might be largely driven by, let's say, low diversity in extreme environments. Like, okay, there is nothing in Sahara, there is very few species in Arctic tundra and so on. So if there is like really strong limit by of course there is low diversity. Yeah? But if you go to, let's say, like moderate resource then maybe like other factors such as terrain heterogeneity or something like that may be
I can see that, and that's a great point. I was talking to some of my colleagues from Brazil, and they were saying, why do we always ask why there's so many species in the tropics? Why don't we ask why there's so few species in the temperate regions? Because from their perspective, the default would be the tropics, and like you were saying, it might be there's some limitations, physiological probably, in the regions which have low richness, rather than asking the other way around. And reframing the question might maybe give us a more complete picture.
In the last study, using those islands, so you showed the effect of phylogenetic structure and the functional structure. You didn't compare those two aspects of biodiversity. Martin did and published last year in his paper and showed that these facets of diversity are usually decoupled. So they are not coupled for plants. It was shown for European grasslands, and probably it is also true for other vegetation types. So did you somehow combine these two aspects of diversity? Because high phylogenetic diversity connected with functional similarity or homogenization could be some refugial effects. On the other one could be some dispersal limitations or something like that.
I think I've run the analyses. I'm not sure I remember the results completely, but we saw that there is a correlation between species diversity and functional diversity, maybe also with the phylogenetic. And it would be interesting to identify, for example, the places where they are decoupled. And now that you say that there is a paper, I should have a look and use it as an inspiration.
Yeah, we were really curious where are those places where the pattern is decoupled. And you spoke about communities. So for you, communities are species pool. So all the species living at that island, but it is not necessary they really compete. Is it true? Because we have community where the species are grow on the same place. So would it affect something?
Absolutely, right? So like even these islands are of different sizes, and like we assume that when the species are on the same island that they interact with each other, like, or maybe eventually they interact, but it is an assumption, right? Potentially strong one. Like again, one argument would be that maybe because we can predict the total number of species on an island, it shows that maybe it's a meaningful unit. Because if it was much more complicated, somehow this stochasticity would prevent us from explaining the variation in island diversity. But it's a very indirect argument, so I completely understand. Subdivided, and eventually there's a critical population size which sets the number of species that can survive in the long term. That's the reason. So I guess maybe at the beginning the competition facilitates speciation, but once you hit the limit of the environment, it might be that it actually suppresses diversity.
And these limits are much lower in the continents which you always have with its stand.
I think so. It seems that the rules of the game really are different on islands and continents. And actually, like much of our theory in ecology and evolution is based on the islands. Like the Galapagos finches, the adaptive radiations. And if it really is the case that these natural laboratories, the islands, play by principally different rules than the continents where most of the species live, this could be something to consider when adjusting the theory that we have.
Thank you very much.
Okay, so thank you very much for this presentation and for this discussion. So let's close this presentation. Thank you once again. And who has some additional questions, so you are invited to come with us to the pub or krug, where the table is booked.